Okay, we're going to start this section with a really big concept. This is a big concept in Chapter 6, and it's a big concept for you overall in general biology. And that is that we need to talk about cell types. So we have eukaryotic cells and prokaryotic cells, or the eukaryotes and the prokaryotes. So the basic structure, function, of cells, that's defined as a cell, but now we're going to subdivide that into two types, prokaryotic and eukaryotic. We have three domains that we talk about in life. We're going to talk about that taxonomy and that hierarchy later in the semester, but we're going to touch on it here to get us started. And only the domains of bacteria and archaea consist within the prokaryotes. All other forms of life, protists, fungi, animals, plants, they're all considered eukaryotic cells or eukaryotes. And so we need to differentiate what's the difference between a prokaryote and a eukaryote. Before we do that differentiation, I want to grab what's common. So we have some pretty basic features of all cells. All cells are going to have a plasma membrane. All cells are going to have a semi-fluid, kind of thick substance contained within them. That fluid is called cytosol. Cytosol will be liquid in its nature. When we talk about cytoplasm, we're talking about the liquid cytosol and then any organelles that are present. So cytosol and cytoplasm are not interchangeable terms, so you do want to know the difference between them. But everything inside the plasma membrane except the nucleus gets its, we'll talk about that in a bit, but the nucleus gets its own exemption to a lot of things. And so everything inside the plasma membrane, with the exception of the nucleus, is considered part of the cytoplasm. Basic features of all cells is that they're going to have chromosomes. They're going to have some form of nucleic acid. Typically we talk about DNA, and then the other thing that all cells are going to have are ribosomes. We'll learn a little bit later that ribosomes are the basically the workbench of the cell where proteins are made. So they're going to be the protein making factories, or the workbench that's in the cytosol. Prokaryotic cells. So with these basic ideas in mind, we need to talk about prokaryotic cells. Prokaryotic cells are characterized in the following ways. They do not have a formal nucleus. They do have a region, an unbound region, a free-floating in the cytoplasm region that seems to be the focal point for the DNA, for the nucleic acid. We call that region the nucleoid. You can see it here in bold. The other thing about prokaryotic cells is they don't have any membrane-bound organelles. So they don't have any organelles that are formally structured or have a membrane around them. And the cytoplasm itself is contained simply by the plasma membrane. So eukaryotic cells have no nucleus. In place of a nucleus, they have a region in the cytoplasm or in the cytosol where the DNA, the nucleic acid, hangs out. That's called a nucleoid, and no other membrane-bound organelles. Okay, eukaryotic cells are going to be characterized a little bit differently. So in a eukaryotic cell, the nuclear DNA will be bound within a nucleus, and a nucleus is a very specialized organelle that has a membranous nuclear envelope around it. Eukaryotic cells will have other membrane-bound organelles, and we'll talk about a bunch of them. And then the cytoplasm in the region between the plasma is contained between the plasma membrane and the nucleus. So there's this exclusion of cytoplasm from the nucleus. Again, that special dispensation that we end up giving the nuclei. Eukaryotic cells are much larger than prokaryotic cells. Some individuals will call them more complex because they have the membrane-bound organelles, but in any instance, however you look at them, they are certainly larger. Remember, I divide my classroom into 1, 3, 5, 2, 4, 6, but for here you guys are just going to make sure that in your notes, in your study methods, you have the ability to adequately compare and contrast cytosol and cytoplasm. And then you want to be able to contrast and compare the prokaryotic and the eukaryotic cells. Okay, so I'm going to do this little bit on plasma membrane, and then we're going to start touring the cell. And the reason I'm going to just put this plasma membrane right here is it's really a single slide for this part of our discussion. And the reason that is simply because we talk about it extensively in Chapter 7. So the plasma membrane is also called a plasmalemma, P-L-A-S-M-A-L-E-M-M-A, plasmalemma. And what it is it's a selective barrier. It allows for the passage of certain things into the cell, and it prevents other things from entering the cell. And it's not just a one-way door. It's not just entry. It's also exit from the cell. So it's like the gatekeeper for the cell. It's the border, it's the fence, it's however you want to look at it, but it defines the edge of the cell, and it's essentially the gatekeeper for anything exiting or entering the cell. It's made up of it's biological, so it's made up of a double layer of phospholipids. You should remember phospholipids from our talk about lipids in general in Chapter 5, and then we'll talk a lot about this double layering factor when we get to Chapter 7. The really truly, and this is you've got to get this now, and it's going to be hit home repeatedly in Chapter 7, is this third bullet point here, that plasma membranes create gradients. They create and they maintain gradients. So there's a disparity between what's inside the cell and what's outside the cell, and that's going to be vital at a later point in time when we start talking about how we're going to transport things in and out, and how if we're going to do it passively or actively, meaning are we going to be able to just diffuse something in and out of the cell, or do we have to actually spend energy or ATP to do it? So I'm going to wrap this one right here, and next we're going to talk about we're going to start with just a basic tour of the eukaryotic cell.